Okay, so experiment 14 was the Flagella experiment, and the purpose was to show how Flagella move a bacteria through a medium. And it is a differential test of flagellated cells versus non-flagellated cells, and so it does allow you to tell the two apart. So how do Flagella move bacteria? So what happens is there's protons in the periplasm that are pumped across this membrane by these MOT proteins. And what happens is, conceptually, you can think of it as the MOT proteins start to spin, and then they're it's like a gear here that because this one's spinning, this starts to spin. And then because this MS ring is spinning, then this whole thing right here is spinning, which then spins the hook, which then spins the flagella. And so that's not what actually happens, but conceptually that's a good way to look at it for the midterm. And so protons are pumped through, and then it spins these things counterclockwise, and it's all because of that proton gradient. Now, you've probably seen this picture like 4 times already by now, but these are the 4 different types of flagella right here. You have monotrichous with one flagella, lophotrichous with multiple flagella on one end, and then these two are your polar flagella. And then you have amphitrichous with one on each end, and then peritrichous with them all over. Okay, so flagella are very small, and so they're hard to stain in and of themselves. Now, in this lab, she did stain some cells that were flagellated that we did get to see underneath the microscope. But primarily what you would do is do a motility test, because those are really easy to do. Motility tests are there's not supposed to be any either, but low agar concentrations, and what that does is it allows the cells to move throughout the medium, and then they have this tetrazonium salt, and what that does is it's an electron acceptor, so it's like oxygen for humans, in that it accepts electrons for metabolism, but in its oxidized form it's colorless and soluble, but in its reduced form, it's red and it precipitates out of solution. And so what that means is that if you have flagella and you stab them in there, they'll use this tetrazonium salt as an energy source to move throughout the medium. And because they have the ability to move throughout this low concentration agar, then you have the ability to produce, and as a result, it makes this red cloud throughout the whole entire thing. If you have an unflagellated cell, they can't move throughout it, and so you just have a red line at the stab line, because they're still going to be using this tetrazonium salt as an electron acceptor, but they can't move, so it's all concentrated on that stab line. And that's pretty much it for that experiment. It was relatively simple, so try to come to an understanding of that.